So the topic of the presentation today is from lab bench to G-EPDs, completing the picture for breed improvement. Now, my role at GeneSeek, and keep in mind I've only been there for about 1.5 years, but I've known GeneSeek for many, many years. And today is actually the first day that I've been at Clay Center. I've been in Nebraska, moved from Canada to Nebraska about 1.5 years ago. This is the first time I'm here. And I tried to make my way here. I was so concerned and trying to be here on time that I forgot to gas up. So some of you might actually need to help me out on the way out of this research center. But anyways, so my role at GeneSeek, I'm Market Development Manager, and I work primarily with the beef breeds in the US and partnering labs in Canada and beef breeds in Latin America and Brazil. They found out I hold the Brazilian citizenship, so I got voluntold to work in that market. But we were able to hire a great team in Brazil, so my work is being shifted towards just hopefully just the US, Canada, and Latin America. Genomic testing allows you to jump ahead of the competition. I love this. So I'm leaving this picture for now. A little bit of about GeneSeek. You toured GeneSeek today. And you may have seen this slide before in previous presentations. Now, we are a market leader in genomic analysis for agribusiness and companion animal industry. We started in Lincoln in 1998 with 1 student intern. And we're presently with, we have 65 full-time employees, 10 PhDs, and 2 DVMs. 15,000 square feet facility. And we're on track to process about 1 million samples by the end of this fiscal year. Some of the services that we provide include high content SNP profiling, market-assisted selection, identity management, and veterinary clinical diagnostics. I apologize, I also have a cold. I was at BIF and I still haven't been able to get rid of that. GeneSeek was acquired by Neogen, and Neogen is our parent company. About 3 years ago, just a little over 3 years ago. And it continues to, it's a small company, comparatively speaking, but it has a big mission, okay? And the mission is to provide DNA services, DNA testing services at the lowest cost possible. So we try to bring genomics to the masses. And one of the things that we do, one of the services that we provide is the right tool for the job, meaning right sizing your genotyping method, custom panels for you. Again, this is just a picture of one of our labs. This is the DNA extraction lab. You guys at the back will probably recognize this. We're on track to process 1 million samples. We are expanding, okay? We are hiring more people. If you guys are interested in working for GeneSeek, they were able to lure me from Canada, cold Canada, to move to Lincoln, Nebraska, the happiest city in the States. It was voted. Did you guys know that? I know. It's great. And here's just a snapshot of some of the genomic solutions that we offer. So starting out with project management, we'll do a little bit of consultation. We'll ask our clients excuse me, what they're interested in, what sort of experiments they like to work on. And we try to provide to them the tools, whether it be 50K, GGPHD, GGPLD, et cetera, that will enable them to complete their research projects, as well as small panels in sequenome, for example. Tissue and hair samples and DNA, we'll take any sample type that you send to us, excuse me, and we'll try to process them on either Illumina or Affymetrix. We have both platforms at GeneSeek. Of course, Illumina, I think Jerry can tell you, everyone is pretty efficient. I think every lab today is pretty efficient in processing Illumina technology because of the type of technology they have. It's amenable to high throughput. Custom content, we have Illumina iSelect. So the GGPHD that you probably have heard of, the GeneSeek Genomic Profiler High Density, or the GeneSeek Genomic Profiler Low Density, those are all custom chips that GeneSeek was able to put together and order from Illumina. The concept there is that you start out with a base, the 7K chip that was a chip that was off the shelf, and you start adding content. Anything else that, genetic abnormalities, coat color, anything you want, you put together your SNP list, and you send it to Illumina. Of course, GeneSeek, because of the purchasing power that we have, we were able to order a lot of these chips and in turn provide them to breed associations for the lowest cost possible. And over to the right, it's sort of, the identity diagnostics platforms that we have. Well, let's focus a little bit more on the high content genotyping. So Matt talked about 50K, GGPHD, GGPLD, and he talked about that being the basis, the behind the scenes for genomic enhanced EPDs today, for all of the breed associations practically. Remember that slide? He showed that table with all of the breed associations working really hard, high numbers of samples on 50K. I mean, this was how you got it done, right? The 50K is a chip processed in Illumina technology. And this is what it looks like. And each of these little guys here is a sample, okay? So when we talk about chip, sometimes people will think, well, one chip, one sample. Well, just figuratively speaking, but in fact, there's 12 or 24 samples on a chip. Sequenome, if you tour the lab today, you went into the Sequenome lab, you saw the mass array, the mass specs, and that's a platform that we use for the lower, smaller panels. So co-color, myostatin, the identity profile, anything that is less than 500 SNPs would be processed in Sequenome. What else I'm thinking of? Some of the genetic abnormalities, NH, they're SNP-based, that would be Sequenome. Anything that has a really large deletion, like AM or CA, that's going to be processed on LightCore technology. This is a bit of an evolution of the bovine genomic tools. So again, Matt talked about the 50K. This was back in, I think, 2006. Somebody correct me. It was commercially available. And then the bovine HD, the Illumina HD, this is 777,000 SNPs. And then later on, BOS1, this is the Affymetrix chip that's also available. And I think it's 640,000 SNPs. The 7K, remember I talked about the 7K being the base content for all of the custom chips are available today, the GGPHD and the GGPLD. So they're all based on using the 7K as the base. Okay. And then, of course, the GGPHD, the GeneSeek Genomic Profiler, high density. This is the concept. Start out with off-the-shelf Illumina chip, and you start adding content. Parentage SNPs, co-color, anything you want. And a lot of the researchers here today have been absolutely instrumental in providing the content for these chips. Okay, so my hat's off to you, all your research, students working on their master's and PhD work, contributing towards this chip, and it's available for you. This is the basis of putting together the GGPHD, the best of the SNPs from the 7K, the 50K, and the 777K. Oops, supposed to have an animation there, but I don't. And this chip has, when it was all said and done, about 77,000 SNPs. And I'll show you later on the slide, it has 28,000 markers in common with the 50K. Now, that's going to be important information for you to understand how we do and how we use this chip today so that we don't lose previous information, previous work that was done on the 50K. Right? A lot of people will ask us, well, if you start out your training population with the 50K and now you're using a chip that only has 28,000 markers, aren't we losing information? Aren't we losing the work that we did before? And the answer is no, because we have tools today that you guys put together that enable imputation from the 28,000 to the 50,000 and creating MBVs from those 50,000 markers. So the SNP effects that Matt talked about. So let's go over a little bit about how that training data set was put together. Now, breed associations, many started building their 50K training data set years and years ago. a lot of research projects have enabled or kind of pushed you guys towards getting more and more of that information so that you can, send that to Dorian, to Jerry, put it all together in one master, I don't want to call it Excel spreadsheet anymore, server, and use that database to come up with genetic predictions, okay? The magical number, I think we said it was 1,000, okay, so that they were able to get, a target number for breed execs and seed stock and producers to help out with getting this deployed. So that was the magical number. Sometimes, and I know for a fact, you're able to come up with good predictions, good correlations, even with less than 1,000, about 100 or 800 or so would be good enough. But that's depending on the breed, and that depends on how well you're able to select the animals part of that training population, right? The higher the accuracy of those animals making part of that training population, the better the correlations are going to be. Now today, many of the all but 2 are using the GeneSeek genomic profiler, either the HD and the LD, and we're going to see why. Now, the official rollout date was May 1st, 2013. And remember that I said that there's 28,000 markers on the GGP HD in common with the 50K, and 9,000 markers from the GGP LD in common with the 50K. Now, the prediction equations were developed using the 50K, right? Remember the question that I posed that most people ask, well, are we losing information when we're moving to this new chip? Excuse me. Now, we need to impute to the 50K, whether you use the GGP HD or the GGP LD, in order for us to use the current prediction equations. And I'll show you the overall system that is in place now at GeneSeek because you guys worked really hard in collecting those samples, sending those in to Dorian, Jerry, Allison, everybody here to come up with those prediction equations. And the result is prediction equations need to be updated with the mapped markers, right? The 50K chip, not all of those markers on that chip have been mapped, you can correct me if I'm wrong, anybody, to a position exactly on the genome. So those ones that are unknown today and were part of that genomic prediction calculation, once we use imputation, we actually have to remove those unmapped markers. And that's okay because the correlations are still up there. We don't lose any information when we do that. Now, not all breeds have switched to the GGP HD. Breeds that have their training, or still in progress of their training population on the 50K, Maine, Adrian, Charolais, we're still building the training population with the 50K. We still have some 50K left in inventory. The reason why we moved to the GGP HD was a price decision. The GGP HD actually costs less for us to purchase than the 50K. So it made more sense for us to switch. And on top of that, you have 50,000 extra markers that could help on the retraining of the population in the future. Okay. I'm going to skip this one because I think. Now, what is imputation? Just a brief overview of imputation. It's a method of determining some genotypes on the computer using actual genotypes on relatives. Right? And it provides options for cheaper routine genotyping. If you have a chip that has a low density, that is of low density, and it's based, if you know that the price of a chip is based on the number of SNPs, you know that that chip is going to be a lot cost effective, I want to say cheaper, cost effective than if you were to purchase something like the GGP HD. Now, you would have to perform imputation. Imagine you've got progeny that was genotyped on a GGP LD, a low density, but you do have information on a sire or dam, and imagine the sire was genotyped using the 50K. You proceed to create haplotypes using that training population, that 50K training population. And this is breed specific, by the way. Okay? I cannot create reference haplotypes for Angus and then trying to predict imputation using Hereford, right? You gotta use the 50K Angus and then use imputation for Angus. I mean, it makes sense. Second slide is to start filling in those gaps, right, from information from relatives. And hopefully you've been able to fill in all of the gaps to the point where it's almost 100% accuracy. That's what imputation. Any questions so far? No. Okay. Now, if you were to remember nothing about this presentation, I'd like you to remember a couple more, slides. And this is the overview. This is sort of the operations today of how the system works at GeneSeek. Training population, breed specific. You helped out your breed association by collecting samples, by going out there, collecting samples from your animals, sending those samples in to us. GeneSeek genotyped those samples on the 50K initially, and now we're in GG-PhD. The raw data is available to all of our clients. We don't hold on to anything. Every client has a share file online, secured. You can log in, you can download your data. You can get the raw genotypes, work with them Saturday morning, add some columns to Excel spreadsheet and work away. Everybody has access to them. We, as well as you and Dorian Garrick, right? So Dr. Dorian Garrick, that spearheads the NBCC initiative to create G-EPDs. He's got access of those as well. And with those data, we create reference haplotypes, breed-specific, and prediction equations. Right? Hopefully you've reached 1,000, and the correlations coming up with those animals that you have, 1,000 or maybe a little bit less, are enough that your breed association will make a decision, or has made a decision, to deploy G-EPDs for the membership. The application of G-EPDs would be as follows. Calf, low accuracy EPD, you would collect a hair sample from that calf, you would send that in. I want to focus a little bit about the hair sample itself. The more samples you send to us, the better, because that allows us to do redos. Right? If the first time we genotype or extract DNA from hair and we don't get a good success rate, by having something to go back to, we're able to extract DNA again, an independent extraction, and then redo that analysis for you and then send that back to the breed association. So the more the merrier, please. And try to collect them early and send those in early to the breed associations. Sometimes we'll see breeders, not you guys, sending samples to us, and I bet you, next day they call in, where are my results? So, but not you guys, I know that. So, sooner the better. Send it to us in advance. If you know you've got a bull sale happening, go out there, collect those samples, send them to us, and we'll do the best we can to turn those results around to you as soon as possible. We are expanding in light of the fact that corporate development keeps acquiring new businesses and integrating them in our current operations. We are expanding, we're hiring people. We are purchasing more equipment so that we can provide faster turnaround of results. So we appreciate your patience with us, and I'm looking at a few people here. In the turnaround time, we're working at it, and we continue to work at it, so thank you. Continuing, genotyping process, GG-PhD, we deliver raw data to the beef breeders, executives, and staff. That goes through the reference haplotypes portion of the process. Okay, that needs to go through first, and then you get virtual genotypes. Remember, the imputation process happens right here, from the 28,000 to the 50,000. And once you've got the virtual genotypes, we send those through the pipeline of prediction equations that is breed specific, so that we can calculate the MBVs from the imputed virtual 50K genotypes. And this next process here is essentially, and we send those to the breed associations, and they in turn work with their genetic evaluation companies to do the good stuff that Matt talked about, all of the procedures and blending or genomic relationship matrix, all of the methodologies currently used so that they can incorporate the MBVs into the National Cattle Evaluation. Does that make sense? No? Okay. Last slide, I'm going to leave you to ponder about whether or not you want to move to Lincoln. This is a picture of us, and I'm happy to say, you can't see me, but this was taken the day, this is 2 years ago, the day that I came to GeneSeek for my first interview, and I'm behind the glass doors, so I'm not, you can't really see me, but I'm in the picture. And you know, go Big Red. I've drunk the red Kool-Aid. So all the way from Brazil, soccer was my favorite sport, and then hockey in Canada, and now football. So I'm all over the place. But you know, Nebraska, the good life. So y'all young leaders looking to make a difference in the beef industry, you know, Dr. John Pollock started out as a lab assistant, right, John? So, you know, look, now he's head of the world.